Polysaccharides.
The acid insoluble pellet also has polysaccharides, carbohydrate as another class of macromolecules. The polysaccharides are long chain of sugars. They are thread, literally a cotton thread, containing different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, that is glucose. Cellulose is a homopolymer. Starch is a variant of this but present as a storehouse of energy in plant tissues. Animals have another variant called glycogen. Inulin is a polymer of fructose. In a polysaccharide chain, say glycogen, the right end is called the reducing end and the left end is called the non-reducing end. It has branches as shown in the form of cartoon figure 9.2, which can be seen later. Starch form helical secondary structures. In fact, starch can hold iodine molecules in the helical portion. The starch I2 is blue in color. Cellulose does not contain complex helices and hence cannot hold iodine. So this is the figure 9.2, which is the diagrammatic representation of a portion of glycogen. So this is the one. Plant cell wall are made up of cellulose. Paper made from plant pulp and cotton fiber is cellulosic. There are more complex polysaccharides in nature. They have a building blocks, amino sugars and chemically modified sugars. Example, glucosamine, N-acetyl, galactosamine, etc. Exoskeleton of arthropods, for example, have a complex polysaccharide called chitin. These complex polysaccharides are mostly homopolymers. Nucleic acid. Another type of macromolecule that one would find in the acid insoluble fraction of any living tissue is the nucleic acid. These are polynucleotides. Together with polysaccharides and polypeptide, these comprise the true macromolecular fraction of any living tissue or cell. For nucleic acids, the building block is a nucleotide. A nucleotide has three structurally distinct components. One is a heterocyclic compound, the second is a monosaccharide, and the third a phosphoric acid or phosphate. As you notice in the figure 9.1, the heterocyclic compounds in nucleic acids are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymine. Adenine and guanine are substituted purines while the rest are substituted pyrimidines. The skeletal heterocyclic ring is called as purine and pyrimidine respectively. The sugar found in polynucleotide is either ribose, a monosaccharide pentose, or 2-deoxyribose. A nucleic acid containing deoxyribose is called deoxyribonucleic acid DNA, while that which contains ribose is called ribonucleic acid RNA. Structure of proteins. Proteins, as mentioned earlier, are heteropolymers containing strings of amino acids structurally of molecules means different things in different contexts. In inorganic chemistry, the structure invariably refers to the molecular formula, example NaCl, MgCl2, etc. Organic chemists always write a two-dimensional view of the molecules while representing the structure of the molecule, example benzene, naphthalene, etc. Physicists conjure up the three-dimensional views of molecular structures while biologists describe the protein structure at four levels. The sequence of amino acids, that is the positional information in a protein, which is the first amino acid, which is the second, and so on, is called the primary structure, which can be seen in the figure 9.3a of a protein. A protein is imagined as a line, the left end represented by the first amino acid and the right end represented by the last amino acid. The first amino acid is also called the N-terminal amino acid. The last amino acid is called the C-terminal amino acid. A protein thread does not exist throughout as an extended rigid rod. The thread is folded in the form of helix, similar to a revolving staircase. Of course, only some portion of the protein thread are arranged in the form of a helix. In proteins, only right-handed helices are observed. The others, other regions of the protein thread are folded into other forms in what is called the secondary structure, which can be seen in the figure 9.3b. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to the tertiary structure, which can be seen in the figure 9.3c. This gives us a three-dimensional view of a protein. The tertiary structure is absolutely necessary for the many biological activities of the protein. Some proteins are an assembly of more than one polypeptide or subunits. The manner in which these individual folded polypeptides or subunits are arranged with respect to each other, example, linear string of spheres, spheres arranged one upon each other in the form of cube or plates, etc., is the architecture of a protein, otherwise called the quaternary structure of a protein. Figure 9.3d. Adult human hemoglobin consists of 4 subunits. Two of these are identical to each other. Hence, two subunits of alpha type and two subunits of beta type together constitute the human hemoglobin HB. Enzymes. Almost all enzymes are proteins. There are some nucleic acids that behave like enzymes. These are called ribozymes. One can depict an enzyme by a line diagram. An enzyme, like any protein, has a primary structure, that is amino acid sequence of the protein. An enzyme, like any protein, has the secondary and tertiary structure. When you look at a tertiary structure upon itself, the chain crisscrosses itself and hence many crevices or pockets are made. One such pocket is the active site. An active site of an enzyme is a crevice or pocket into which the substrate fits. Thus, enzymes through their active site catalyze reactions at a high rate. Enzyme catalyses differ from inorganic catalysts in many ways, but one major difference needs mention. Inorganic catalysts work efficiently at high temperatures and high pressures, while enzymes get damaged at high temperature, say about 40 degrees Celsius. However, enzymes isolated from the organisms who normally live under extremely high temperatures, example, hot vents and sulfur springs, are stable and retain their catalytic power even at high temperatures up to 80 to 90 degrees Celsius. Thermal stability is thus an important quality of such enzymes isolated from the thermophilic organisms. Chemical reactions. How do we understand these enzymes? Let us first understand a chemical reaction. Chemical compounds undergo two types of changes. A physical change simply refers to a change in shape without breaking of bonds. This is a physical process. Another physical process is a change in state of matter. When ice melts into water or when water becomes a vapor, these are also physical processes. However, when bonds are broken and new bonds are formed during transformation, this will be called a chemical reaction. For example, BaOH twice plus H2SO4 gives BaSO4 plus 2H2O is an inorganic chemical reaction. Similarly, hydrolysis of starch into glucose is an organic chemical reaction. Rate of a physical or chemical processes refers to the amount of products formed per unit time. It can be expressed as rate is equal to delta P divided by delta T or dP divided by dt. Rate can also be called velocity if the direction is specified. Rates of physical and chemical processes are influenced by temperature among other factors. A general rule of thumb is that a rate doubles or decreases by half for every 10 degree Celsius change in either direction. Catalyzed reactions proceed at rates vastly higher than that of the uncatalyzed ones. When enzyme catalyzed reactions are observed, the rate would be vastly higher than the same but uncatalyzed reaction. For example, CO2, carbon dioxide plus H2O, water, in reversible manner in the presence of the enzyme carbonic anhydrase, H2CO3, which is carbonic acid, is formed. In the absence of any enzyme, this reaction is very slow, with about 200 molecules of H2CO3 being formed in an hour. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction speeds dramatically with about 6 lakh molecules being formed every second. The enzyme has accelerated the reaction rate by about 10 million times. The power of enzymes is incredible indeed. There are thousands of types of enzymes, each catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction, when each of the steps is catalyzed by the same enzyme complex or different enzymes, is called a metabolic pathway. For example, glucose gives 2-pyruvic acid. C6H12O6 plus O2 gives 2C3H4O3 plus 2H2O. Is actually a metabolic pathway in which glucose becomes pyruvic acid through 10 different enzyme catalyzed metabolic reactions. When you study respiration in chapter 12, you will study these reactions. At this stage, you should know that this very metabolic pathway with one or two additional reactions gives rise to the variety of metabolic end products. In our skeletal muscles, under anaerobic conditions, lactic acid is formed. Under normal aerobic conditions, pyruvic acid is formed. In yeast, during fermentation, the same pathway leads to the production of ethanol, which is alcohol. Hence, in different conditions, different products are possible. How do enzymes bring about such high rates of chemical conversions? To understand this, we should study enzyme a little more. We have already understood the idea of an active site. The chemical or metabolic conversion refers to a reaction. The chemical which is converted into a product is called a substrate. Hence, enzyme, that is protein with three-dimensional structures including an active site, convert a substrate S into a product P. Symbolically, this can be depicted as S gives P. It is now understood that the substrate S has to bind the enzyme at its active site within a given cleft or pocket. The substrate has to diffuse towards the active site. There is thus an obligatory formation as a transient phenomenon. During the state where substrate is bound to the enzyme active site, a new structure of the substrate called a transition state structure is formed. Very soon after the expected bond breaking or making is completed, the product is released from the active site. In other words, the structure of substrate gets transformed into the structure of products. The pathway of this transformation must go through the so-called transition state structure. There could be many more altered structural states between the stable substrate and the product. Implicit in this statement is the fact that all other intermediate structural states are unstable. Stability is something related to energy status of the molecule or the structure. Hence, when we look at this pictorially through a graph, it looks like something as in the figure 9.4. Here the figure 9.4, the concept of activation energy can be seen in this picture. Here we can see a graph in which the x-axis is represented by the progress of the reaction and the y-axis is represented by the potential energy. So this is the level of potential energy of the substrate and this is the level of potential energy of the product. And here we can see a high potential energy state which represents the potential energy state of the transition state. And here we can see the energy level of the substrate in this manner, and from this line to this portion, that is from the substrate energy level to the transition energy level, is called the activation energy. Here we can see two activation energies. with mentioned as the highest one is activation energy without enzyme and the lowest one we can see as activation energy with enzyme. This portion can be deal with in details in the later sessions. So continuation, the y-axis represents the potential energy content. The x-axis represents the progression of the structural formation or states through the transition state. You would notice two things, the energy level difference between S and P. If the P is at a lower level than S, the reaction is an exothermic reaction. One need not supply energy by heating in order to form the product. However, whether it is an exothermic or spontaneous reaction or an endothermic or energy requiring reaction, the S has to go through a much higher state or a transition state. The difference in average energy content from that of this transition state is called activation energy. Enzymes eventually bring down this energy barrier, making the transition of S to P more easy. Nature of enzyme action. Each enzyme E has a substrate S binding site in its molecule so that a highly reactive enzyme substrate complex ES is produced. This complex is short-lived and dissociates into its products P and the unchanged enzyme with an intermediate formation of the enzyme product complex EP. The formation of the ES complex is essential for catalysis. E plus S, ES gives, EP gives E plus P. The catalytic cycle of an enzyme action can be described in the following steps. Step 1. The first. The substrate binds to the active site of the enzyme fitting into the active site. Second step. The binding of the substrate induces the enzyme to alter its shape fitting more tightly around the substrate. Step 3. The active site of the enzyme, now in close proximity of the substrate, breaks the chemical bond of the substrate and the new enzyme product complex is formed. And the step 4, the enzyme releases the product of the reaction and the free enzyme is ready to bind to another molecule of the substrate and runs through the catalytic cycle once again. Factors affecting enzyme activity. The activity of an enzyme can be affected by a change in the conditions which can alter the tertiary structure of the protein. These include temperature, pH, change in substrate concentration, or binding specific chemicals that regulate its activity. Temperature and pH. Enzymes generally function in a narrow range of temperature and pH, which can be seen in the figure 9.5. Each enzyme shows its highest activity at a particular temperature and pH called the optimum temperature and optimum pH. Activity declines both below and above the optimum value. Low temperature preserves the enzyme in a temporarily inactive state, whereas the high temperature destroys enzymatic activity because proteins are denatured by heat. Concentration of substrate. With the increase in substrate concentration, the velocity of the enzymatic reactions rises at first. The reaction ultimately reaches a maximum velocity Vmax, which is not exceeded by any further rise in concentration of the substrate. This is because the enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules, there are no free enzyme molecule to bind with the additional substrate molecule, which can be seen in the figure 9.5. This is the figure 9.5, and here we can see the effect of change in A represents the pH and B represents the temperature and C concentration of substrate on enzyme activity. Here we can see Vmax and Vmax by 2 and corresponding Km value, and this can be dealt in details in the later sessions. The activity of an enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme. When the binding of the chemical shuts off enzyme activity, the process is called inhibition and the chemical is called an inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme, it is known as competitive inhibitor. Due to its close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Consequently, the substrate cannot bind, and as a result the enzyme action declines. Consequently, the substrate cannot bind, and as a result the enzyme action declines. Example: inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in structure. Such competitive inhibitors are often used in the control of bacterial pathogens. Classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on the type of reaction they catalyze. The enzymes are divided into 6 classes, each with 4 to 13 subclasses, and named according by a 4-digit number. Oxidoreductases or dehydrogenases. Enzyme which catalyze oxidoreduction between two substrate S and S dash. Example: S reduced plus S dash oxidized gives S oxidized plus S dash reduced. Transferases. Enzymes catalyzing a transfer of a group G other than hydrogen between a pair of substrate S and S dash. Example: S single bond G plus S dash gives S plus S dash G. S bonded with G plus S dash gives S plus S dash bonded with G. Hydrolysis. Enzyme catalyzing hydrolysis of ester, either peptide, glycosidic, carbon-carbon single bond halide, or PN bonds. Lysis. Hydrolysis. Enzyme catalyzing hydrolysis of ester, either peptide, glycosidic, carbon-carbon halide, or PN bonds. Lysis. Enzymes that catalyze the removal of group from substrates by mechanism other than hydrolysis, leaving double bond. Here we can see that carbon bonded with X, that carbon is again bonded with Y, that is gives X bonded with Y plus carbon double bonded with another carbon. Isomerases includes all enzymes catalyzing interconversions of optical, geometric, or positional isomers. Ligases. Enzyme catalyzing the linking together of two compounds. Example: enzyme which catalyze joining of CO, CS, CN, PO, etc. bonds. Cofactors. Enzymes are composed of one or several polypeptide chains. However, there are a number of cases in which non-protein constituents called cofactor are bound to the enzyme to make the enzyme catalytically active. In these instances, the protein portion of the enzyme is called the apoenzyme. The 3 kinds of cofactors may be identified: prosthetic groups, coenzymes, and metal ions. Prosthetic groups are organic compounds and are distinguished from other cofactors in that they are tightly bound to the apoenzyme. For example, in peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and hydrogen, heme is the prosthetic group and it is the part of the active site of the enzyme. Coenzymes are also organic compounds, but their association with the apoenzyme is only transient, usually occurring during the course of catalysis. Furthermore, coenzymes serve as a cofactor in a number of different enzyme catalyzed reactions. The essential chemical components of many coenzymes are vitamins. Example: coenzyme nicotinamide and adenine dinucleotide NAD and NADP contain the vitamin niacin. A number of enzymes require metal ion for their activity, which form coordination bonds with side chains at the active site and at the same time form one or more coordinate bond with the substrate. Example: zinc is a cofactor for the proteolytic enzyme carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of the enzyme. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of the enzyme. Summary. Although there is a bewildering diversity of living organisms, their chemical composition and metabolic reaction appear to be remarkably similar. The elemental composition of living tissue and non-living matter appears also to be similar when analyzed qualitatively. However, a closer examination reveals that the relative abundance of carbon, hydrogen, and oxygen is higher in living systems when compared to inanimate matter. The most abundant chemical in living organisms is water. There are thousands of small molecular weight less than 1000 Dalton biomolecules. Amino acids, monosaccharide and disaccharide sugars, fatty acid, glycerol, nucleotides, nucleosides and nitrogen bases are some of the organic compounds seen in living organisms. There are 20 types of amino acids and 5 types of nucleotides. There are 20 types of amino acids and 5 types of nucleotides. Fats and oils are glycerides in which fatty acids are esterified to glycerol. Phospholipid containing addition of phosphorylate nitrogenous compound. Only 3 types of macromolecules, that is protein, nucleic acid and polysaccharides, are found in living systems. Lipids, because of their association with membranes, separate in the macromolecular fraction. Biomacromolecules are polymers. They are made of building blocks which are different. Protein are heteropolymers made of amino acids. Nucleic acids, RNA and DNA are composed of nucleotides. Biomacromolecules have a hierarchy of structures: primary, secondary, tertiary and quaternary. Nucleic acid serves as a genetic material. Polysaccharides are components of cell wall in plant, fungi and also of the exoskeleton of arthropods. They also are storage forms of energy, example starch and glycogen. Proteins serve a variety of cellular functions. Many of them are enzymes. Some are antibodies, some are receptors, some are hormones and some others are structural proteins. Collagen is the most abundant protein in animal world, and the ribulose bisphosphate carboxylase oxygenase rubisco is the most abundant protein in the whole of the biosphere. Enzymes are proteins which catalyze the biochemical reactions in the cells. Ribozymes are nucleic acids with catalytic power. Proteinaceous enzymes exhibit substrate specifically require optimum temperature and pH for maximal activity. They are denatured at high temperatures. Enzymes lower activation energy of the reactions and hence greatly the rate of the reactions. Nucleic acids carry hereditary information and are passed on from parental generation to progeny. So that's all for today's lesson. And one more thing, if you like this video very useful, then like, share and subscribe, and also comment your toughest chapter that you want to be like this in the next video. So keep going, keep study well. Thank you so much. Thank you.